I have begun talking about, in the vertebrates, I've been talking about metabolism again. I've been talking about whether animals are ectothermic and endothermic. And I want to explain a little bit about vertebrate anatomy, circulatory anatomy, and the heart anatomy, and how that directly relates to whether an animal can be endothermic or not. So you may recall, to be endothermic, an animal has to be able to deliver a large volume of blood in a relatively short period of time. That blood needs to be under high pressure going to the tissues. And then also, it has to have a very high oxygen content. And so I want to start just by quickly reviewing just blood flow in general. So if we look at this hierarchy, again, blood, if we start with the heart, there are two types of heart chambers. There's ventricles and atria. And so if we have blood in a heart ventricle, when that ventricle contracts, that would force blood out of the heart. And the blood would then move into what we call arteries. So arteries are very thick-walled blood vessels that can tolerate the pressure being generated by that ventricular contraction. Arteries then begin to branch into smaller vessels, which would be arterioles, still relatively thick-walled, that can tolerate that pressure. And then they continue to branch into vessels that have smaller diameters, but the vessel wall thickness gets less and less, and those would be the capillaries. And so the capillaries have the thinnest walls, but also the smallest diameters in individual vessels. And that's where the gas, nutrient, and waste exchange occurs. So that's where oxygen goes to the tissues, carbon dioxide into the bloodstream, and nutrients into the tissues, wastes from tissues into the bloodstream. So all of that exchange occurs in the capillaries. Then blood would begin its journey back to the heart, in effect. It would now begin to flow into venules, and then, which are small veins, and then veins, which are larger. And then veins eventually will dump back into the heart, and the heart chamber that receives blood is known as an atrium. And then when the atrium contracts, it will drive blood into the ventricle and start the process over. So ventricular contraction to arteries, to arterioles, capillaries, venules, veins, back to the atrium, and then to the ventricle again. All right, so I want to look first at this interface between the arterioles and venules and the capillaries. So as we look at blood flow then, again, we're going to have blood flowing from arterioles through capillaries. And these capillaries could either be in gills, they could be in lungs, but they could also be in tissues. And then eventually into venules that will take the blood back to the heart. So here again, blood would be coming away from the heart, goes through the capillary bed, and then back to the heart. Now, again, looking at the capillaries, what's important about them is that the vessel thickness is very, very thin, just a single cell layer, so that gas, nutrients, and waste can diffuse in and out freely. And the individual vessel diameter is very small. But if we were to compare, say, an arteriole vessel diameter to the sum total of the vessel diameters of all of the capillaries here, I hope you get a feel for the fact that the total capillary vessel diameter is actually greater than the arteriole that's leading into that. So what's important about this is it relates to this pressure concept. So if you imagine in our own anatomy, there's a very large blood vessel called the aorta that comes out of our heart. It has a diameter of maybe the diameter of a nickel, maybe a quarter, something like that. And eventually that will branch into a variety of different capillaries. And if you were to take the cross-sectional diameter of all of those capillaries, it might be as big as the studio I'm in. It would be huge. But the point is, if you have a pipe, with this diameter, and then you have another pipe with a much larger diameter, and you're moving the same amount of blood or fluid through those pipes, the smaller pipe will have a much greater pressure. The larger pipe will have a lower pressure if you're moving the same volume of fluid through the two pipes. So it's the same idea as we look at our capillary bed then. Blood that's in the arterioles will be under fairly high pressure. And then as that blood moves through the capillaries, that pressure is dissipated because the total cross-sectional area is so much greater than the vessels leading in. And so then the blood going into the veins is under very low pressure. So this is something that we have to consider in a couple of minutes when we talk about animals that are ectothermic and endothermic. And the pressure is so low, as a matter of fact, that when you look at the structure of veins, veins actually have valves in the blood vessels to prevent the backflow of blood. So whereas arterioles and arteries, have the force of the ventricle, the heart ventricle forcing the blood out, and that's what's driving blood through the arteries and arterioles, once the blood passes through a capillary bed, there's virtually no pressure. It's almost zero pressure. And so it's really movement of the animal. In our cases, it's movement, muscular movement that will help milk that blood eventually back to the heart. Okay, so as we look at fishes, amphibians, reptiles, and mammals and birds, let's see how their anatomy relates to whether they can be ectothermic or endothermic. So you may recall that the osteichthyes have a two-chambered heart. They're ectothermic. And so as we look at their heart anatomy, then they have one atrium and one ventricle. Those are considered the two chambers. So when an atrium contracts, it drives blood into the ventricle. When the ventricle contracts, it will send the blood to the gills, and there's a capillary bed in the gills. Now, blood's going to the gills under fairly high pressure. That's a good thing, but then that pressure is dissipated in the capillaries of the gills. It's good to send the blood to the gills because it can be oxygenated, and then from there you're going to send the blood to what we call the systemic circulation, which would be all of the tissues, the muscles and so on. It's what we're most interested in here. And then the blood gets deoxygenated there and eventually returned back to the heart. So you send it to the gills to get oxygenated and then to the tissues. That's good. But here's the part of the problem with this anatomy is that you're sending blood to the gills, running it through a capillary bed, and all of your pressure is dissipated there. And then you're sending it to the tissue capillaries. So it's kind of unusual in that we're actually going, instead of arterioles to capillaries to veins, we're actually going, arterioles to capillaries to venules, back to capillaries, and eventually back to veins again. So fish then cannot be endothermic. They're ectothermic because they don't meet our criteria. So can they move a tremendous volume of blood? Not so much, because the pressure is dissipated by this first capillary bed. So they can't really send a lot of blood rapidly through the body. So they can't really move a large volume of blood. Can they send it under high oxygen concentration? Well, sort of, but they are living in water. So water doesn't have as high an oxygen concentration as air, so it's fairly high, but even that may not always be the case. And then thirdly, can they send it under high pressure to the tissues? And the problem is no, because the pressure gets dissipated in the gills, and so they cannot send this blood to the tissues under high pressure and force that oxygen into the tissues. Now, let's look at amphibians. So amphibian heart is a three-chambered heart, but amphibians are still considered ectothermic. And so as we look at the amphibian heart, then we have two atria and one ventricle. And you'll notice that the right atrium will receive blood from the general circulation or systemic circulation, and that means coming from the muscles basically. And then that blood will go into the right atrium and it gets pumped into the ventricle. That blood then gets sent to the lungs. So that's deoxygenated blood. It gets sent to the lungs. It will be oxygenated in the lungs. And I should also mention here that in amphibians, that would also be the skin circulation. So they would also be sent going to the skin. So it's lung and skin. And then that blood is oxygenated, is coming back to the left atrium, and then sent into the only ventricle. So there's one ventricle here. And notice that when this ventricle contracts, that will also send blood to the general circulation or systemic circulation. So that would go out to the muscles. So the situation here is that we are sending blood to the lungs to be oxygenated and then out to the tissues once it's oxygenated. But since there's only one ventricle, this creates a couple of problems. One, we're having mixing of oxygenated and deoxygenated blood in the ventricle. So we're not ever sending out fully oxygenated blood to the tissues. So that's one limitation of this system. And then also, in terms of pressure, the capillaries that line the lungs, remember, are going to be next to air space. And so if you were to crank up the pressure here, those capillaries basically would expand. They would balloon up and eventually rupture potentially, and you'd bleed out into the lungs. Whereas if you look at capillaries that are found in the general circulation, those capillaries are surrounded by other tissues. So if you do increase the pressure in your muscle capillaries, for example, you have muscle tissue around them so that it can help support that capillary. So what limits the amount of pressure that the ventricle can send blood to the tissues under, it's limited by the amount of pressure you can send blood to the lungs, which would be much less than what you can send blood to the tissues. So, so as a result, amphibians are also ectothermic because, again, if we look at our three criteria, can they move a large volume of blood? They can move a pretty good volume of blood. So that's not the biggest problem for them. Are they sending highly oxygenated blood to the tissues? Not so much, because they're mixing oxygenated and deoxygenated blood. And can they send that blood to the tissues under very high pressure? No, because they have to send blood to the lungs under low pressure, because if they send it under high pressure to the lungs, the capillaries will rupture. So they have to send blood to the tissues under relatively low pressure. So bottom line in both fishes and amphibians is that they cannot deliver enough oxygen to support endothermy. So I'll talk about reptiles and mammals in another supplement.